Starting point for understanding everything, and as we presented it, the first thing we looked at was the internal capsule, and then we looked at the basal ganglia and the thalamus. So here's the interventricular foramen right here. These are the fibers of the fornix, and this is the corpus callosum, and there would be a covering right here called septum pellucidum that's now gone. But here's the transition from the lateral ventricle out here in the hemisphere to the third ventricle right here. Okay, and here's that interventricular foramen. When you look at it, that interventricular foramen, the thing we identified was it's at the point of the anterior thalamic nucleus. It's at a point where it's just above the uncus. It's at a point where the internal capsule comes to the ventricular line, or right there at the genu of the internal capsule. And then streaming away from that, define the anterior and posterior limbs of the internal capsule and the relationship to the putamen, globus pallidus, caudate, and thalamus. Okay? So what we want to do is use that as a landmark. Well, when the interventricular foramen, when the fornix fibers and all that are gone, we don't really have a good landmark to do it, so we have to use something else that was related to that, and that was the anterior pole of the thalamus. So when we look at this, I hope this brain works okay. When we look at this, here is the anterior pole of the thalamus right here. And kind of see how this groove is right here. If we go inferior down to the perihippocampal gyrus, you see right here this bump of the perihippocampal gyrus. So that's the uncus. And that was posterior cerebral artery that I just removed. Okay? So here's perihippocampal gyrus right here. And it narrows right here behind the corpus callosum, which is kind of torn up, and then becomes the cingulate gyrus, and the cingulate gyrus runs all the way around like this. Part way back on the cingulate gyrus, there's this branch that comes up here, which is that marginal branch, and defines this paracentral lobule. So that's the sensory and motor part of the homunculus for the lower extremity contralaterally. Okay? So that's where that is. So to make this horizontal cut that then how we used in that template to define the internal capsule, what I want to do is be able to cut through that interventricular foramen. And since I don't have a good marker for that, I'm going to use this anterior pole of the thalamus. Okay? And I'm going to put the knife there, and then I'm going to cut through the splenium of the corpus callosum. And what you notice is this V-shaped structure. Okay, so there's the internal capsule, and then outside of that is the putamen. Here's the posterior limb of the internal capsule, and medial to the posterior limb of the internal capsule is the thalamus, and the thalamus comes to the third ventricle surface. The anterior limb of the internal capsule, medial to that, is the head of the caudate nucleus, and it pushes into the lateral ventricle. Okay? And all of these are little branches of the middle cerebral artery. Okay? Now what we want to do then is to make a cut inferior to that, and what we want to be able to see with respect to that as we move down through the perihippocampal gyrus to the uncus is we want to be able to see the transition from the hippocampal formation, which has its presence about this length, and it's coiled inside of the perihippocampal gyrus. And then at the uncus, inside of that is the amygdaloid complex. So the combination of those two are where we consolidate memories. The periapicampal gyrus is where we consolidate more explicit memories, and then the amygdaloid complex is where we consolidate more emotional memories. Okay, so the anxiety, fear of spiders kinds of things, really more fearful types of memories. Okay, so the first cut about 1 centimeter down. Ooh, sweet.
You seem excited about that.
There is something really exciting. Okay, so a bunch of things on this. So the first thing, our target was to really look at the hippocampal formation lying inside of the periapicampal gyrus. And you can see kind of this coiled structure right here. So that's the hippocampal formation. Okay? But what this cut did was it actually cut through a couple of different structures. You can see right here are these fibers. If we go back to this, here's our internal capsule right here. And as those fibers funnel down, they ultimately become the cerebral cortex, or cerebral peduncle, right there. So that's cerebral peduncle has all of the fibers that are leaving cortex that are going to the pons, to the bulbar cranial nerve nuclei, and to the spinal cord. And just inside of that, you see this darkened line of cells is substantia nigra pars compacta. Okay? The other thing that this did is you see here is the optic tract coming back, and as it comes back, it gets to this golder area right here as the lateral geniculate nucleus. And from the lateral geniculate nucleus, you see this wide, heavy band of white fibers going back here are the geniculocalcrine fibers, or the optic radiations. Okay?
Cool.
What you don't see, because we're still above the uncus, which is right here, is you don't see the amygdala. So if we make one more cut,
That looks like a really awesome cake cutter.
Wow.
Seriously?
I'm not eating cake now. Thanks. I thought on video, really.
I think it's a little bit exciting.
Well, I'm never going to eat cake now.
so here was the uncus, here's that bump, and you see deep to that is this large homogeneous area, there's no definition to it, is the amygdaloid complex. And behind it you see this squiggly area right here is the very rostral end of the hippocampal formation. Okay? The other thing that you can see is this long strip of fibers right here, running back like that. Those are the inferior most parts of those geniculocalcrine fibers, which are the Myers loop fibers. So that's those fibers right there. Okay? So if we put this all back together, we should be able to then go through this at each level and kind of follow those structures. The next thing we want to do is we want to be able to take a coronal cut that then somewhat mirrors slide 10. And again, we're going to start at that interventricular foramen, or since we don't know where that is, we're going to start at the anterior pole of the thalamus. So again, here's another brain. Again, septum pellucidum isn't there. We'll get rid of anterior cerebral artery. Rid of that. So here's the anterior pole of the thalamus. If we go in here and pull off internal carotid artery, here's the uncus, or that bump, that rostralmost bump of periapicampal gyrus. Here's periapicampal gyrus coming around to become cingulate gyrus. Here's the marginal sulcus paracentral lobule.